Okay, so we're recording. And for questions, if you have questions, just type in the chat box. You can just put sort of a general description of your question, or you just say, I've got a question. And then at the end of the talk, I'll call on you to unmute yourself and you can just ask the question yourself. If we run out of time and there's still more questions, then the speaker can actually, if they can understand what the question is, just type their response. So feel free to put as much detail into your question as you want, and we'll go from there. So with that, I'm going to turn it over to Felipe and his mentor to do a brief introduction. So have at it, guys.
All right, good morning. My name is Allison Bean. I'm an assistant professor in the Department of Physical Medicine and Rehabilitation. I work in the Ambrosio lab under the mentorship of Fabrisia Ambrosio. It's my distinct pleasure to have the opportunity to introduce Felipe Mola Curi. So Felipe is a rising senior at Upper St. Clair High School. He's hoping to study neuroscience as an undergraduate and then attend medical school afterwards. Just in terms of the lab this summer, it's been an absolute pleasure to work with Felipe. He's extremely intelligent and a fast learner, but what's kind of stood out to me is just his incredible curiosity. He's constantly asking excellent questions, and it's certainly kept me on my toes. He's also really just a joy to be around. He fits seamlessly into the lab, and so many people remarked about how surprised they were to find out that he was a high school student and not already an undergraduate. It's been really great to see him grow over the summer. He has really great technical skills, and he's also just extremely driven. We were doing a lot of new techniques that I hadn't done before, and despite some setbacks, he was always eager to try to find solutions and try again. I'm definitely going to miss working with him every day, but I'm hoping I can convince him to come back to the lab in the future. And with that, I will go ahead and turn it over to Felipe.
Thank you for the introduction, Dr. Bean. Good morning, everyone. Today, I'm very excited to share with you the project that I've been working on for the past 7 weeks on how neuromuscular electrical stimulation affects extracellular vesicle characteristics in the murine model of aging. So skeletal muscle is crucial for us because it allows us to move and also protects our organs. However, as we age, we lose muscle mass and also muscle strength alongside our ability to regenerate muscle tissue after injury. All of this combined kind of leads us to a loss of mobility and increased disability amongst elders. Novel treatments to restore muscle function are needed in order to better quality of life amongst an older generation. So it's been pretty well known that exercise, more specifically resistance exercise, can improve muscle function. There have been studies suggesting that exercise can help us maintain a more youthful level of tissue function and also other systemic effects such as muscle function, neurogenesis, cardiac function, and tissue regeneration. In order to further understand how these systematic effects occur, there have been studies using a technique called heterochronic parapiosis, in which they sew an old mouse and a young mouse together and combine their circulatory systems in order to exchange blood. And through these experiments, they have found that the old mouse does gain a more youthful phenotype due to this experiment, and they have similar results to exercise. And this has brought us to wonder whether exercise can improve muscle function by signaling molecules inside of the blood. One way that researchers have recently identified extracellular vesicles as a potential way of delivering these circulatory factors in cells and tissue. Extracellular vesicles are lipid bilayer nanoparticles, and they contain lipids, proteins, and genetic material such as RNA and DNA. These particles are secreted by cells and tissue, and they're inside of the blood, meaning that they're very important mediators for intercellular communication. Recent studies in our lab has proven that EVs do have rejuvenating properties. So in this experiment, as you can see on the top picture, this is a cross-section of the tibialis anterior muscle of a normal young mouse. And you can see how the muscle fibers stained in laminin are very similar in size and shape. However, once they stain the injured old muscle, you can see that it does not look anything like it. And the cross-section decreased in size. However, once they injected a serum with young extracellular vesicles, it kind of gained some of the same properties back to the young muscle, and the cross-section increased. We do know that this is due to the extracellular vesicles because once they injected the same exact serum, however, they didn't have any EVs in them, they did not see any regenerative properties, and the muscle just looked the exact same as injured old muscle. Now, returning to the previous discussion on how exercise can improve skeletal muscle function, we wondered if the positive effects of EVs could be further increased through exercise. However, since mice can't do squats on the spot, we decided to use neuromuscular electrical stimulation, also known as NMES, to passively induce repetitive muscle contraction. Our hypothesis was that NMES will restore a more youthful phenotype to circulating EVs and enhance skeletal muscle regeneration. So as a first step, we wanted to determine the optimal NMES regimen to stimulate EVs. For this, we had 5 different groups. First, we had a young untreated and an aged untreated group that were our controls. We also had an acute muscle contraction group with 1 session of NMES and 2 chronic groups with 5 sessions of NMES, and 1 was sustained or euthanized 3 hours later, and the other was euthanized 24 hours later. All of our NMES regimens were the same for all of our NMES groups, in which we did 2 sets of 10 muscle contractions on each hind limb for a total of 20 contractions, and there was a 5-minute break in between. And we had 5 seconds on and 10 seconds off. We decided to pick these groups because we wanted to first compare chronic versus acute muscle contractions. So that's why we had the 1 versus 5. And there have been studies showing that EVs can only sustain changes after like 3 hours after the last procedure. So we wanted to see if that were true. So we wanted to see if the changes would sustain after 3 hours or 24 hours. Once all of the groups were euthanized, we took their blood through cardiac puncture and spun it down so the red blood cells could separate from the serum. Once we took out the serum, we used size exclusion chromatography to isolate the extracellular vesicles. And with those extracellular vesicles, we did 2 different things. One, we did EV characterization so that we could assess for size and concentration. And we also did an in vitro experiment in which we added the 5 samples to muscle stem cells that were already old, and we tested them for myogenic potential. So for our EV characterization, we did have to pull all of our samples together, thus we were unable to assess for any statistical significance. However, our preliminary results showed that the group with 5 sessions of NMES euthanized 24 hours after the last session did have the greatest concentration. And we did also find that the EV particle diameter decreased as we did more sessions of NMES, and it became closer in size to the young control. So for our in vitro experiment, we wanted to do an immunofluorescent staining for PAX7 expression. PAX7 is a marker for muscle progenitor cells. And as you can see, the green is PAX7 and the blue is stained with DAPI. So that's nucleus. And whenever you see the overlap, that just means that it's a living cell that's PAX7 positive. There was no statistical significance in this data because, or this may have been due to difficulty getting our cells to adhere to our slides, and we did lose a few cells. So we will have to repeat these studies to see if the data remains reproducible. We did do the same thing with MyoD, which is an early marker for myogenic potential. And it's very similar to our previous pictures with the pink right here. You can see it a little bit better. The pink being the MyoD and the blue being the nuclei stained with DAPI, and whenever there's an overlap, they're positive for both. Again, there's no statistical significance due to the cell detachment, but we will be doing these experiments again. So to summarize our results, EVs had a smaller diameter in NMES groups and young untreated samples compared to our aged controls. Our EV concentration was highest with chronic NMES and euthanized 24 hours later. And there was no real significance between PAC7 and MyoD in those expressions and between the groups. And we do believe that that was due to cell detachment, but we'll be taking a closer look at that in previous experiments. So our main conclusion is that chronic repetitive muscle stimulation may alter EV characteristics in aged animals. So in the near future, we do hope to repeat the immunofluorescence stainings for MyoD and PAC7. However, we will be coding our slides in fibronectin in order to make these cells appear a little better so we can have a larger sample size and they can be healthier when we do that. We also want to increase our sample size so our statistical analysis can be a little better and we have more data. Furthermore, we do want to investigate changes in myogenic differentiation, and we'll be doing that by staining for myosin heavy chain and desmin. So we'll be looking at that too. And lastly, we would like to do an in vivo experiment by injecting EVs into injured skeletal muscle, and we would be using a mouse model for that. So we'll be doing that soon too. So for my acknowledgements, I wanted to first thank the entire Hillman Academy leadership team, and specifically Dr. Boone, Mr. Lifshitz, Dr. Duncan, Dr. Alexander, and the Hillman Cancer Center. Thank you for this opportunity. I really appreciate it. I really did enjoy my time here. And so yeah, thank you for all the support and everything you guys did for us, from lectures to all the help you supported us with. I also wanted to thank Okay, there we go. I also wanted to thank the entire Ambrosio lab, but also more specifically, Dr. Ambrosio, Sunitha, Kai, Hirotaka, Amanda, Zach, and Shruti. You are all so welcoming to me when I came to the lab and let me shadow you with your experiments and help me with mine, taught me so much. So I'll forever be grateful for all of you. So thank you for that. I also wanted to thank my Hillman Academy peers, even though we didn't get to see each other as much as I would have hoped. I still always look forward to our meetings on Fridays to hear about your experiments and what you guys have been up to. So thank you for all of your support. And most importantly, I want to thank Dr. Allison Bean. You were truly the best mentor I could have asked for this year. You not only made everything very interesting and helped me with all of our experiments and giving me all the support, but you also made my summer a lot more fun than I would have expected for doing research. So thank you for that. And if you guys have any questions, you can go ahead and ask them now.
Awesome. Good job, Felipe.
Thank you.
You could, I guess, go ahead and stop sharing and maybe we can see everybody then. Does anybody have questions? I'll kick it off. You talked about, so you do these stimulations and even you've got two different stimulation regimens. One is the kind of short one, and even in the longest time, you do 5 stimulations and then harvest after 24 hours. Mm-hmm. But I wonder, is that long enough? Like, what if you did stimulations every day for a month or 6 months? Is there anything to say that, you need like a much longer term of stimulations to change the quantity or the quality of the EVs?
There could be more changes if we did longer, but we could look at that in the future, but I don't think there's anything showing right now. There have been previous studies that show that this is long enough to do some changes, but we can take a look at that in the future too.
Got it, got it. Anybody else have a hard question? I'll ask another question. What's known about, so you showed even after your 5 stimulations at the 24-hour time point, and I get it that you had to pool the samples, but you had more EVs in that longer treatment. But what about quantity of EVs versus quality of the EVs, like the cargo that's in there? Is there anything known about how that might change over time or with stimulation?
So that's actually a really good question. We did talk about that, but we need to measure the protein using BCA, and we're going to be doing that soon. We tried to, and my BCA didn't really work out as well as I hoped. So we'll be doing that soon. And we also want to do an RNA sequence later. So we can measure for all of that at some point as well.
Got it. Got it. That's awesome. If anybody has questions that come to you over the next while, you can put them into the chat and Felipe can ask them.
Felipe, I've got a question.
Yeah.
This is Will Concrete. I collaborate with Dr. Ambrosio's lab over at the Neuromuscular Research Lab. Great job in the presentation. That was really, really well done, and exciting to see some of the work that you and Allison have been doing. In terms of, as you know, the EVs that you assessed in circulation are a heterogeneous population. They're coming from a lot of different cell sources. Have you all considered looking at any origin markers of where these EVs are coming from and analyzing those specifically?
Oh, first of all, thank you. No, not yet, but we do hope to do that in the future. First, we wanted to make sure that we get our immunofluorescence stains done and we get those in vivo experiments. But once we do all of that, we do look to take a closer look at everything else as well.
Okay, let's go ahead and move on. Thank you, Felipe. That was awesome.
Thank you.
And next up, we have Maddie Douglas.
Hello. Can you guys hear me?
Yep, we hear you.
Okay. Yeah, so hi guys, my name is Steven. I'd like to introduce Madeline Douglas. She's a rising senior from Woodland Hills High School. She has interest in neuroscience, computer science, genomics, and aging. Her plans are to apply for graduate school programs when she has completed her high school degree. I'd like to say that it's been an absolute pleasure hosting Madeline this summer within our lab. She is truly a wonderful student. And we've been really impressed with the way that she's been able to adapt to performing the research and understanding the literature in such a short period of time. I'm personally very impressed with how, especially within the last 2 weeks, that Madeline was able to balance her research responsibilities while also attending band practice during the day for her fall semester. So in typical grad student fashion, potentially taking on more than they can handle. But in any case, we're very excited for the work that she has performed during the summer. And yeah, I'll let Madeline take it away.
So hi, my name is Madeline Douglas. And as you have heard, this summer I worked in the Kozai lab under my mentor, Stephen Wellman. And today we are looking at the spatiotemporal characterization of novel CX3, CR1, CSPG4 cell population around chronically implanted neural interfaces. Now, that was a lot of information. So before we get into the biological happenings, we need to step back and consider, why does this matter? Well, implanted microelectrode arrays, such as the Michigan array and the Utah array that you see on the left, allows us to more effectively study the brain's physiological properties and answer significant neuroscience questions. This knowledge then allows us to manipulate the brain more effectively or even lessen the impacts of neurodegenerative disorders. But not only that, neurotechnology has the ability to provide this. A woman with quadriplegia, a form of paralysis, eating a chocolate bar with a robotic arm and hand that she is controlling with her brain. Now, we want these devices to last as long as possible for patients suffering from neurodegenerative disorders. However, this ideal situation is not happening. The performance of the device degrades over time, a degradation believed to be due to the harsh foreign body response to the electrode. In a study that used the Utah array, the device with the various rows of probes, on monkeys for an extended period of time, they observed only about 60% of the channels out of the 100 electrode array that was still able to detect neuronal units within the first few months. And as years passed, only about 20% of the electrode channels are viable. This is because of the foreign body response to the electrode. The insertion of the implants causes ruptured blood vessels, the activation of immune cells, and the encapsulation of the electrode, all which contribute to the decline in the performance of the device. However, it is not entirely known what part of the foreign body response is causing these declines. But here we see two figures of GFP microglia on the left and DS-red parasites on the right. These two scavenger cells are really important for the neuroimmune responses in the brain and occupy various spatial regions in the brain, with microglia occupying the neuronal space with the function of maintaining neuronal networks and injury repair, and the parasites tending to cover blood vessels to regulate blood flow and maintain homeostasis within the body. These cells, though the variability of the immune cells is poorly understood, are expected to be near the inserted probes. However, we have seen a new population of cells appear around the implanted device, separate from the microglia and the parasite populations, that is simultaneously expressing both GFP and DS-red fluorophores, meaning they express both the promoters of microglia and parasites. We were not previously aware of these cells, so in order to develop strategies to mitigate and treat their responses, we need to characterize the behaviors of these cells. So ultimately, we're trying to learn more about these dual localized cells. That means we want to understand the cellular behaviors of these CX301 CSPG4 cells around in response to the implantation of the foreign device. So we are looking at the appearance, distribution, and fate of these cells. When post-insertion do they start appearing? Is it specific to the electrode? What distance are they from the electrode? We are characterizing the migratory behavior and motility of these cells. How fast are they moving? At what time post-insertion are they the most active? And we're also looking at the surface coverage of these cells. Do these cells participate in electrode encapsulation? What time post-insertion does surface coverage peak? And what proportion of encapsulation is the microglia, the parasites, or these dual localized cells? So using a dual fluorescent mouse model, or a mouse model that expressed both markers of GFP microglia and DS-red parasites, we implanted microelectrode arrays within the cortex and employed 2-photon microscopy to track cellular dynamics in vivo over 28 days post-insertion. To determine the appearance and distribution of the CX301 CSPG4 cells, we counted the cells and created a binary mask of the electrode shanks by using a rotated stack, such as the one on the left, and then outlining the probes to create a mask like on the right. Then a custom MATLAB script was performed the distance transformation on the cells with respect to the electrode. So next we performed 2 more analyses. To determine the migratory behavior and motility of the CX301 CSPG4 cells, the movement of the processes in cell bodies was tracked using the measure function in ImageJ. To then determine the percentage of surface coverage of the CX301 CSPG4 cells, the microglia, and the parasites cells on the electrode shanks, the average Z projection was performed on rotated stacks, and each channel was then thresholded using the ISO data feature in ImageJ, and a custom MATLAB script was then used to determine the percentages and proportions of each of the cells. So here we can see the results of tracking the co-localized cell distance. As we can see, there are no cells counted on day 0 and day 1. However, we can see on day 4, we start to see the appearance of these cells. Now, it is important to note that these cells are not seen on the contralateral part of the brain. So this means we can speculate that the injury is a result of the implant itself. As a result, as time passes, despite the increase of cell totals, we can see in the bottom chart that the percentages of cells within the 0 to 20 micron range increases, suggesting the cells are moving towards or forming closer to the electrode. With this in mind, we start to examine the motility of these dual localized cells. So these cells demonstrated active process extension and peaked about at an average of 6.41 microns a minute 1 week post-implantation. And from 7 days post-insertion to 28 days post-insertion, the average speed of the processes overall declined. Although on day 21, there was a high amount of variability between the speeds of the electrode. And so as expected, there is a great amount of GFP coverage on the first day due to injury. However, based on a 2A NOVA, no statistically significant difference in the dual localized cell coverage over time and in comparison to coverage from the CX3 and CSPG4 cells, which are just the microglia and parasites, was found. But we are still able to discover new characteristics despite this. We can see that the DS-RED GFP coverage is happening, meaning the cells are participating in encapsulation. Now, because we are seeing these cells are participating in encapsulation, we are able to understand they are demonstrating behavior typical of reactive cells rather than just passive cells. We can also see from the way that the cells migrate and encapsulate the electrode that they are participating in the foreign body response to these implants. And although it is not significant, we can see the data is trending towards significance, meaning possibly adding more animals to the study or excluding the outlier with a lot of bleeding. There could be a significance then, but that would be a study for another time. So ultimately, we understand that these CX301 CSPG4 cells start appearing about 4 days post-insertion within 70 microns of the electrode. The cell movement of these cells peaked about 7 days post-insertion, and the DS-RED GFP coverage participates the most in electrode encapsulation, but there's no statistical significance to that. However, the DS-RED GFP, even being there, means that they are participating in this foreign body response. So future work would include the continuation of the study of these dual localized cells. That would include learning about what type of cells are these dual localized cells coming from, to eventually facilitate the development of more advanced neural implant designs that react more positively to the brain upon insertion, as well as being able to develop innovative therapeutic strategies to mitigate this neurodegeneration and tissue failure following brain injury or disease. And I would like to give a special thank you to my mentor, Stephen Wellman. He has made this summer quite possibly one of my best summers I've ever had. I'd also like to thank my PI, Dr. Takashi Kozai. An additional thank you to Dr. Boone and Solomon Lifshitz from the Hillman Academy, and a very special thank you to the Bionic Lab and my TDX site, who made me feel incredibly welcome this summer. And now, if there are any questions, go ahead and ask. Thank you.
Thanks, Madeline. That was excellent. Who has some questions? We just have a shy bunch today.
I can ask the questions.
Okay.
Hi, excellent talk. So you focused on these two cell types, and it was really cool to see the microglial cell moving. Do you think there are other cells moving also in addition to the microglial cells?
Yeah, there definitely are other cells moving. So the parasites are moving, the microglia are moving, and then we also see these dual localized cells moving. So, and we're only in the picture, in the image, we only tracked the microglia and the parasites. So there could definitely be other cells that just weren't shown that are moving as well.
Yeah, really cool. Thinking about immune cells, I guess microglia are immune cells of the brain in some way, but it would be interesting to see if there are additional cells that are playing a role in the response.
Yeah, there definitely are. If I'm not mistaken, oligodendrocytes are definitely participating in this foreign body response, and other types of immune cells like astrocytes are participating in this response as well.
Madeline, and the humans and patients who are having this foreign body response, do you see those same cell types encapsulating the devices?
Yeah, so we haven't tracked that yet. We just looked at the mice, but I would expect the same cells to appear.
Right. I didn't know if maybe those were cells that had been observed in humans, so that's why you track those in the mice.
Yeah.
Maybe so.
Yeah, it's ultimately we're studying to eventually apply to humans, so it definitely would be. We're definitely looking at the same type of cells.
Anyone else? All right. Well, thanks so much. That was really excellent. All right, we're going to move on. Next speaker is Griffin Hurt.
I deserve to introduce Griffin. Sorry for the delay, having
That's okay.
difficulties. So, Griffin is a rising senior at Yale High School, and we had a great time. And it was a pleasure to have Griffin in our lab remotely during this summer. However, we're fortunate to do a few social events, and I know Griffin came to the lab to actually see Matt performing the experiments, so that was really cool. Griffin has excellent programming skills and very good critical skills. He's interested in pursuing a degree in computer science. So I think the project you will see presenting is a great fit. And
Okay, it looks like he has stalled out. Give him another couple of seconds to see if he comes back in. All right.
He is away across. He's in a different hemisphere right now, so that could be part of the part of the problem. But well, I think different hemisphere. So I guess with that, I will just start my presentation. Thank you very much, Dr. Zervantonakis, for the introduction. I don't know if you can hear me, but thank you. Hi, everybody. My name is Griffin Hurt, and this is my presentation on comparing classical image thresholding and machine learning for quantitative cell analysis. So let's talk about the significance of image analysis a little bit. It's a term that's thrown around a lot and talked about in the media a lot, but well, specifically with our image analysis, well, obviously the ultimate goal is for breast cancer research. We want to develop better drugs that are more effective across a larger range of tumor types. And the way that we do that is by getting a lot of data about how well certain drugs work. And the fastest and quickest way of doing that is with image analysis. Specifically in our lab, we want to do quantitative analysis of cells. So we want to do what's called a cytotoxicity assay, which basically addresses how many live and dead cells there are over time. That's researching what in our lab is called fibroblast-mediated drug resistance. So there are cells called fibroblasts that will interact with tumor cells and the drug that we use to treat HER2-positive breast cancer, and that's called lapatinib. And when a lot of fibroblasts are present in the cell, there will be present in the area that will actually decrease the ability of the drug to treat the cancer. And so we want to develop a mathematical model for that interaction. And the easiest way to do that is by training the model with accurate temporal data. And we do that with image analysis. And there are a lot of other biological experiments that require quantitative cell analysis, doing other drug evaluations and migration assays, which look at where cells, if cells are moving properly. So the goals of this specifically was to develop an automated pipeline for analyzing cytotoxicity assay images. So we take a bunch of images of cells with a microscope over time, and then we want to put them into the software program and get a count of living and dead cells with a sort of a time point. We will also want to explore our options for image analysis, their classical methods, machine learning methods, and some neural networks as well. And we also want to compare the benefits and detriments for each option. So the general trend is that the more accurate your final results are, the more computational complexity comes along with it. So if you want more accurate results, you have to sort of live with the analysis taking a longer period of time. So with that, let's get into the two softwares that we compared. So on the left, we have Cell Profiler, which is an open source image analysis software that was released in 2005. It's fairly popular. It's been cited more than 6,000 times at a rate of roughly 1,000 per year. A paper not too long ago rated it top in both usability and functionality among 13 different free image analysis softwares. And it uses classical image analysis techniques. So it uses what's called thresholding, where basically you look at every pixel in the image and you try and draw a line where the brightness represents a cell and where the brightness represents background. So it's a very simple method of basically optimizing this function of splitting the data. And it doesn't require training examples, which is unlike machine learning. But in some cases, it has difficulty identifying cells, which is why we can't just use it everywhere. Whenever cells die, they tend to shrivel up. And we have a fluorescence marker in the cell. So when they shrivel up, they have a very high fluorescence. And so we get some false positives with the thresholding. And then on the right, we have our machine learning candidate, which is Elastic. And it's newer than Cell Profiler. It was originally published in 2011. And one thing that's really nice about Elastic is that it has two separate workflows for both pixel and object classification. So determining what is a cell and determining what is a living and dead cell. And it's not a manually set pipeline like Cell Profiler. So it sort of figures out how to best split the data so you don't have to set it. It uses what's called a random forest classifier machine learning, and it requires manually classified data. So here, over here to the right of the slide, we see this is what's called a decision tree. So a decision tree is able to make a final decision on an input based on several different variables. So as you can see, you basically just step down this tree. So this is the survival of passengers on the Titanic. If they're male, if their age is less than 9.5, then they died most likely. And so what a random forest is it's a decision tree, but you have 100 decision trees and they're all trained on slightly different amounts of data in different variables. So that's the sort of machine learning background here. So now let's take a look at how Cell Profiler does on an actual data set. So over here we have an animation of some cells over time. We can see that cells are dying more over time, which is what we want to see. But we also see some slight issues, which is cells tend to blink in and out of existence. So if you watch this little cluster here, you can see that those cells sort of blink in and out. And so over here to the right, I have an example of where it's missed right here. There should be more cells identified that it's missed. And then here, there's a lot more cells that it's missed. Those dead cells that we talked about, it has trouble classifying. It's missed this one. There's some more right here that it missed. So it does an all right job, but it's not the greatest. It does do a good job at showing the general trends that we're looking for, though. So here's some quantitative output on the EFM 192 cell line, which is a type of tumor cell. So when we look at the trends with no drug, we can see that the cells tend to grow over time. The blue line represents the total viable cells, while the red line represents the dead cells in the image. And so this is, both of these are with 4,000 tumor cells and 4,000 fibroblasts. So we're not really looking at how fibroblasts affect the data in this case. But as you can see, the blue line tends to go up and the red line stays down. But when we give it some drug, 3.0 micromolar of lapatinib, we see the viable cells tends to go down, and the red line, which is dead cells, tends to increase. So more cells are dying when we give it drug. And so now we can look at how Cell Profiler does on ground truth images, which we sort of looked at, but it wasn't as quantitative as this is. So here, any place where you see green clusters of pixels, those are manually identified cells in the image. So that we went through and manually classified where there are cells in the image, sort of like training data for machine learning. And so pure green represents where there are manually identified cells, but cell profiler didn't identify a cell, and red represents where cell profiler identified a cell, and yellow is the overlap. So any place where you see yellow, that means it's good, that means it did a good job. So over on this left image, you can see that cell profiler has done a very bad job at identifying these cells. We see a lot of green splotches, but not a lot of yellow. We see some false positives. Here in the middle, we've seen it's done a little bit better of a job, but we get some of these false positives right here. And over on the right, it's probably done the best of the three. And so overall, that gives cell profiler 61.17% precision. It's got a low false positive rate, which means that generally, if there's not a cell somewhere, it won't think there's a cell somewhere. But it's got a pretty high false negative rate, which means that it's missing a lot of cells, 58.67%. And so here's how Elastic does on the same data set. So we can see that there's a lot less of cells popping in and out of existence, especially in the same cluster we just looked at, where it does a pretty good job at tracking those. But the most clear issue with this is that not a lot of cells are dying, even though cell profiler thought that the cells were dying. In this image, it doesn't look like the cells are dying over time, even though they are, right? So here's an example of it's done a very good job here at making out almost every nucleus of the cell. And here are a lot of dead cells that have been classified as living, which is incorrect. And so here's Elastic, its output on the EFM192 cell line, the same one we looked at with cell profiler, same exact conditions. With no lapatinib, we see that the cells tend to increase over time, which is what we're looking for, and that the dead cell count doesn't change. But with the drug, we see that the cell count doesn't, the cell count is straight, which is what we're looking for, but the number of dead cells does not increase over time, which is a problem. So, but a thing of note is that this line is straight. It doesn't have a downward trend like it did with cell profiler, which is positive because that means that when cells die, we don't stop detecting them. We still detect them, but we're not classifying them as dead, which is a problem. And so here's some Elastic comparison to ground truth. So on the left, we have the image that cell profiler really struggled with, but here we see a lot more yellow in this main cluster, which is a good sign, which means that we're getting less false negatives. And then in the middle, we see where Elastic tends to struggle, which is we have these sort of red areas, which are areas that we got a false positive. So there's no cell here, but Elastic thought there was a cell here. And then on the right, we see it's done a pretty good job here. So we've seen an increase in precision up from 61% to 65%, a slight increase in the false positive rate, 2.51%. And we've seen the false negative rate actually drop below 50%. So that means that more than 50% of cell pixels in these images are being identified correctly. So now let's talk a little bit about the computational complexity of each one of these algorithms. So time complexity is a concept in computer science that indicates how the number of operations required to complete an algorithm changes with a change to the input. And that's also known as big O notation. So with O2 thresholding, which is a common type of thresholding used in cell profiler, it runs in time O(n plus L, where n represents the number of pixels and L represents the number of gray levels, which remains constant at 256 usually throughout these experiments. And then we also have the time complexity for building a random forest, which this would be the training of the Elastic. And this runs incredibly slow. This is mkn log squared n, where n represents the number of pixels, k represents the number of variables, roughly 110 in these experiments, and m represents the total number of trees. And by default, which was in these experiments, it's 100 trees in Elastic. And then predictions with the random forest in its best case scenario runs OM log n, which is just m's your number of trees, n's your number of pixels. And in its best case, it runs m log n, which is a lot faster than O2 thresholding. But in its worst case, it runs in OM times n. So if we look at this graph over here to the right of the slide, we can see this is a log chart of theoretical algorithm operations to the size of the image. So this will be a 500 by 500 image, 1000 by 1000. And you can see that while the best case random forest is very low, the average case, which is the green line, is significantly higher than O2 in every case. So that means that O2 on average, which is classical image thresholding techniques, even the most complex ones, generally run faster than the random forest classifier on average. And this is shown in the experimental computational analysis as well. So on average, we were seeing Elastic takes 10.87 seconds to analyze a 1392 by 1040 pixel image, while Cell Profiler only took about 3.02 seconds. And so that means that Cell Profiler processed each image 3.6 times faster than Elastic, which is sort of representative of what we looked at with the theoretical analysis. So in summary, machine learning methods are able to distinguish cells from the background with greater accuracy than classical image techniques with even small amounts of training data, because we only had about 5 images in the training data set, but yet we were able to see an increase in precision, which is positive. So we saw 65.2 precision detecting cells versus the 61% in Cell Profiler. But with that, the trade-off was greater computational complexity. We saw a increase of almost or greater than 3 times as long for each image. And then classical image analysis techniques are able to classify cells better without training. So it doesn't require any training, but still it was able to better classify cells. And this is sort of representative of an even bigger issue with machine learning, where if your machine learning training data is limited or incorrect, which, like I said, we only had about 5 training images in this situation, it really struggles to classify correctly in contrast to if you have a dead cell fluorescence marker, which is what we had, then Cell Profiler can make it out very easily. So here are just some charts showing how the false positive rate, the false negative rate, and the precision was for each one of the ground truth images we compared, how it did between the 2 softwares. So looking towards the future, what can we do to sort of get a better pipeline for this? Well, we can combine the power of both methods. We can use the machine learning to identify what is a cell, and then use classical image analysis to classify the cell as living or dead. And then the other option that we have is to look into convolutional neural networks. They've been shown to be promising for nuclei identification. However, they do require extensive computation, 2 to 5 times longer per image than Cell Profiler. So you can see it would, in its worst case, it could take even longer than the random forest classifier, and significantly more training data is needed than the random forest classifier. But with that, we get an increase in accuracy. We get a 12.9% increase in accuracy with the compared against Cell Profiler, which is relatively good. So I'd like to acknowledge everybody who's helped me with this project. Thank you to Dr. Ioannis Zervantonakis, he was the principal investigator. Thank you to my mentor, Mr. Matt Poskus, and thank you to Dr. David Boone, who ran the Hillman program ran this great Hillman program and gave us all an opportunity to learn more about biology research. And here are my citations, and thank you very much.
Great job, Griffin. That was excellent. We can have time for 1 or 2 questions. Well, I'll start. How would you improve the Elastic? What would be the next step to make it, to try to get it to actually recognize the dying cells?
So the #1 best way to improve it would just be to give it more training data and classify that training data better. Part of the issue is that I was the one who was actually creating the training data, and obviously I'm not the most qualified to identify what is a live and dead cell just solely based on the images. And so I think that having better and greater amounts of training data would be the quickest way to improve the accuracy of the network.
Got it. Got it. Anybody else? All right. Well, if anything pops in your mind, put a question in the chat. Thank you, Griffin. That was excellent.
Yep. Thank you, everybody.
All right. So next up, we have Marie.
Hey everybody, before Marie starts, I wanted to give her a quick introduction. My name's Ron Fortunato. I was her mentor, and I'm a graduate student in Dr. Spandan Maiti's lab. I had the pleasure of working with Marie over the past couple months. She's an extremely well motivated and comprehensive learner. and an integral part of the research team. She's a student athlete in softball, and that provides a full schedule of travel and games during the summer. And she continued to find time to fully dive into her research project. I think she has a bright future ahead of her. She was looking into colleges like Rochester, New York, that she was excited about. I think she's performed at a very high level and level of professionalism that I think she'll have no problem moving forward in future endeavors, whether that be research or industry data science. She was kind of interested in. I don't want to take any time away from her presentation. So Marie, whenever you're ready, you can continue. Thank you.
Hi, everyone. So I'm Marie Kinchington, and I worked with Ronald to look at computational modeling of aortic aneurysm tissue, and we specifically looked at the biomechanics. So what are some of the characteristics of aortic aneurysms? Aneurysms are caused by a weakening in the wall of the aorta, where the blood pressure increases. As it increases, the wall in the aorta begins to bulge outward. And this often causes, the diameter to increase. And here is a really great representation of the diameter increasing. So here we have a normal aorta at the bottom, about 3 centimeters. And once it gets larger than 5 centimeters, the aorta can actually rupture. And this generally, once the aorta ruptures or dissects, it results in death almost 80% of the time, even with immediate treatment. So what are some of the repairs to the aortic rupture? So you can actually fix an ascending aortic with an ascending aortic replacement, which is where you use a graft to repair the aneurysm. However, this is not always 100% foolproof, and it is generally an elective procedure with patients. So if this is not done, if the graft is not done, it will actually result in aortic rupture or an aortic dissection. So we analyzed 3 different types of tissue from the aorta. We analyzed a TAV tissue, a tricuspid aortic valve tissue, a BAV tissue, bicuspid aortic valve, and as well as a control tissue, which came from the tricuspid aortic valve and in a patient that did not have an aneurysm. We analyzed these tissue in 2 different directions, so a circumferential direction and a longitudinal. Circumferential is going around the width of the aorta, so looking at it crosswise, where the longitudinal direction is going along the aorta, along the aortic tissue. So how do we analyze this data? We analyzed it using a stress stretch graph, and a stress is shown on the y-axis. So the stress is the amount of force that is put to separate the tissue. So when the tissue is separated, the stress goes up. And as the stress goes up, it will change the stretch, which is the change in the shape and the system of the tissue. So we used alpha and beta values to fit the model to an experimental curve. And an alpha value is the beginning to the middle part of the slope. So we changed the alpha using the this little slide right here, and that is going to change the beginning to the middle part of the curve. And we used a beta value, which is going to change the middle to the ending part of the curve. And both of these change different parts in the tissue. So alpha is related to the elastin fibers and beta is related to the collagen fibers. So when we were analyzing this data on a stretch curve, we mainly focused on the failure point. So the failure point is where the tissue reaches its max amount of peak stress. So right here, we can see the failure point happens at B, and we can also see it correlates right here where the tissue begins to break. Here I added the alpha is changing the beginning part of the curve. So from the beginning to point A. And the beta value is going to be changing the ending part of the curve until the failure point from A to B. And this is going to be, the alpha is going to be the elastin fibers changing, and the beta is going to be the collagen fibers breaking. So once we get to point B, the tissue begins to break, and then the collagen fibers also begin to break. So what were our goals? We wanted to further understand the statistics of the 3 different types of aortic tissue, specifically using stress graphs and data tables. We did this putting the data tables into box plots, which we will get to soon. And some of the questions we wanted to answer are either group of aneurysm patients, TAV or BAV, more at risk for an aortic rupture, and can we predict when and where an aneurysm patient's aortic will rupture using a stress graph. So what was the process? So first, we used the graphs to calculate the alpha and beta values. For each patient in each cohort, we had 8 patients in each type of tissue group. Then we used the data to determine the failure point from the graphs. And then we used computational modeling in Python to analyze the statistics of each cohort, such as the means, the averages, the medians, such like that. Then we wanted to compare each box plot for each direction, so circumferential and long, with each other. And we wanted to answer, again, are either group of aneurysm patients, TAV or BAV, more at risk for aortic rupture. Eventually, we wanted to normalize this data for each cohort to predict where other patients' tissue will fail. So here are our results. So first, we have the alpha value, and we can see that they are statistically different, but only in the long direction. So here we have the alpha in the circ direction on the left, alpha in the long direction on the right. And there is no statistical difference in the circumferential direction, which shows that going the width of the aortic tissue, there is no significant difference in the elastin fibers. However, going in the long direction for the TAV versus BAV, there is a significant difference in the elastin fibers going the length of the tissue. Next, we have the beta values. And using these graphs, we could see that the beta values show no statistical significance in either direction. This is really kind of interesting because it showed us that there wasn't a main difference in the bulk of the collagen fibers once the aortic tissue started to break. So although the spreads are different, there is no statistical difference for either direction. Now, most important, we have the peak stress. So the peak stress is going to show us where the tissue is going to fail for each of these groups. We can see that there is statistical difference in both sides, so the circumferential and the long direction. And we can see that in the BAV groups, the peak stress is higher in both the circumferential and the long direction, which really indicated to us where the tissue strengths differ. So in summary, we can see that a higher peak stress means higher stretch, which generally correlates to a higher strength. This means that the probability of rupture or dissection is going to be lower. And because of the peak stress graphs, the BAV tissue is biomechanically stronger because it can withstand a higher peak stress than the TAV tissue. So some of the future directions that we wanted to take was to analyze a BAV control, not just a TAV control, and this was going to help us in comparing the BAV to another control value, which we couldn't do it with the TAV because they're 2 different types of tissue. Next, we wanted to use the means of each kind of tissue to predict the future outcome of patients, which we did not get to do because we didn't have time, but we would like to do that in the future. Okay, so there's a couple of people who I would like to thank. I want to thank Ronald, of course. He's been so helpful. And even though we've been online and I haven't been able to see him in person, he's always been super, super helpful with any questions, anything I have. He's taught me so much about Python. And I told him that I've learned more in 8 weeks than I did in 2 years of coding classes. And then, of course, I want to thank Dr. Maiti, who really helped facilitate my understanding of the TAV versus the BAV and the aneurysms. And of course, I want to thank Dr. Duncan for always being so comfortable with us speaking and during the research roundtables and everything. And Dr. Boone and Mr. Lifshitz, who pretty much helped us all through the Hillman Academy.
Marie, that was outstanding. Thank you.
Thank you.
Who's got some questions? I thought Felipe was going to ask something for a second.
I am. I did have a quick question about the your results. So you said that part of them were significant, but part of them weren't, correct?
Yes.
Like the long one was significant. Okay. Could you use that to go and like advance your studies, or will you have to like, redo the experiments again since part of it was not significant?
So we could do both. If we wanted to redo the study just to make sure it is the results turn out the same, to make sure we can generalize them, but they actually do not correlate with each other. So if one is significant and one is not, that is still okay, because they are measured in different parts of the different like sides of the tissue.
Okay, thank you. That makes sense.
Marie, to what extent is having an N of 8 patients limiting? I mean, typically when people are talking about diversity within humans, you need many, many more N. In mouse studies, 8's pretty good, but what do you think you would need to really get truly, not that your data is not believable, but what would really strengthen that? What kind of N do you think you would need?
Right, so that's a good question. I definitely think you can always use more patients, and always having more patients is going to be better to normalize and generalize the data. However, I think that we had a really good focus on just our 8 patients, because we did not have very many outliers when we did the data tables and the box plots. So that's a really good question though.
And how do you envision using what you're learning here to help someone? I can think of some ways it might work, but I wonder how from an expert like you, how you think it would really work.
Right. So I actually talked with Ronald and Dr. Maddie about this. And so using the peak stress, we can determine what is the amount of, we can correlate that to blood pressure stress that comes from the aorta. And then as the blood pressure stress increases, we can see where the tissue will expand to peak stress. And once it gets to the peak stress, it fails. And then we can kind of generalize that to patients with different kind of aneurysms, because we can see, depending on their blood pressure, where their aorta might rupture.
So then you would need to know blood pressure, and would you need to have some imaging done on their heart prior to that?
Yeah,
right.
So you would use like an MRI slices, and you would put them together, and then you would be able to see the aneurysm that way.
Got it. That's cool.
I have one more question going off what Dr. Duncan said. So you guys said that there are only 8 patients, and I might have missed this, but since there are no outliers, I don't know, did you get like, how different were the ages, like sex, backgrounds, all of that kind of stuff? Or was it all from like more of a control group?
So that's a really good question. I am not 100% sure about that, because this data was taken from the University of Pittsburgh Medical Center, and it was patients who were electively undergoing in thoracic aortic replacement. So we don't know the age or the sex of these patients, but we do have their data.
Okay, thank you.
Okay, if there are no other questions, we can go ahead and take a break. The first 4 talks today were excellent. Thanks to all the speakers and all the mentors for coming in and doing the introductions. We are gonna pause until 10:45, and then Myka is gonna give her talk at 10:45. So yeah, with that, I'll see you guys in about 10 to 12 minutes. Thank you. Welcome back, everybody. Hope you had a good 10-minute break or so. Next up, I guess we still have, well, just a few seconds, so we'll go ahead and proceed. Next up, we have Myka, and I will invite Andre to do the introduction.
Yes, it's my pleasure to introduce Myka Lichius. So Myka attends Oakland Catholic High School and she's starting her junior year. So Myka before already participated in a few scientific programs. So she attended Pittsburgh Regional Science Fair and Pennsylvania Junior Academy of Science in her 7th and 8th grade. And she's also a famed scholar. So I just want to say I was really impressed by Myka's performance during these 6 weeks, what she spent in my laboratory, how she performs experiments, how she works, how she can discuss like different scientific topics, how she understands very well. So you talk to her like a colleague, not like a student. So I'm really looking forward to her presentation and her future scientific endeavor. So I think she has a really bright future in front of her.
Hello, my name is Myka Lichius, and my mentor is Dr. Andre Parhico, and we are located in the Parhico lab at the Aging Institute in Bridgeside Point 1. The title of our project was Methionine Metabolism, Methyltransferases, and Stress Resistance. Key information for our project is that methionine is an amino acid that is obtained from food. Methyltransferases use the methyl group from SAM, which is produced from methionine. Part of it is used for protein synthesis, and part of it is used for metabolic processes in the body. Methyltransferases transfer methyl groups to different molecules like proteins, nucleic acids, metabolites, and they also regulate multiple processes. Methionine restriction is very important because it has multiple health benefits, such as extending lifespan, suppressing cancer, and preventing obesity. And Drosophila is the scientific name for the fruit flies that I am using as a model. So for methionine goes towards creating proteins, and it contributes to different metabolic pathways. For polyamines, they interact with molecules such as DNA and RNA, and changes in levels of these polyamines can result in disease and illnesses in most cases. Cysteine is an amino acid, and it is also a precursor for an antioxidant. SAM is a methyl donor that is involved in transfers to DNA, proteins, and etc. SAM is needed for methyltransferases, and methionine is used to create SAM. There are 150 methyltransferases found in flies. Methyltransferases take methyl groups from SAM and transfer them to their targets. For example, histone methyltransferases transfer methyl groups to histone proteins, and they regulate gene expression. Our hypothesis is that out of the 150 methyltransferases, which methyltransferases mediate the effects of methionine metabolism on stress resistance? For Drosophila, we have to discuss genetic drivers. These genetic drivers are genetic elements to manipulate gene expression in specific tissue. These drivers can be used in either the whole body of the fly or in specific organs to manipulate genetic expression in the different types of flies. Temperature regulation can also be used to deactivate or activate certain drivers. Drosophila is studied because it is easy to focus on the methyltransferases effects, therefore the phenotype is easy to measure. So to study how phenotypes are affected and to test the flies for stress resistance, we have to start a cross of phenotypes. For a cross example, we had a suppressor GAL 80TS, which in 18 degrees Celsius is active and the driver is inactive. In 29 degrees Celsius, the suppressor is inactive and the driver is active again. We combine this with RNA interference, or RNAi, which is driven by a genetic driver, which inactivates a particular methyltransferase. To discuss more about RNAi, it is using the same specific code of RNA already present to double back and turn it off or cancel it out. We use these to test the effects of tubulin Gal4, which is the common driver. For our cross, we had a female with tubulin Gal4 and Gal ADTS times a male with the specific RNAi code. For our experiment, we had set up a cross and completed it at 18 degrees. We then waited a month for progeny and switched the flies to 29 degrees Celsius for 7 days to activate the genetic driver, which then drives the expression of RNAi to turn it off one of the methyltransferases. We then sorted the flies by the balancer for the ones that are wanted. We created the food for the stress assays, then counted the dead flies to compare survival between different groups. Here we have the male rate of survival and the female rate of survival. On the y-axis is the percentage of survival in the flies. Every day, a certain number of flies would die. Our goal is to compare survival between control flies and the ones where we turned off the methyltransferases. We compared the survival separately for males and females because of different features. I will first walk you through the males. In red, you can see the control flies. And then with the yellow line, that gene expression has a shorter lifespan. The orange line lives longer than the others. And then here we tested only 4 different methyltransferases. If you look on the females, the results are different, which would be expected because methionine restriction has different effects between males and females. Here I showed you the results of the one stress assay, which completely depleted methionine from the food. Methionine restriction creates stress for flies since methionine is a necessary component for survival. We also had completed other tests for other stress conditions where we starved the flies, only giving them water, or we would give them a strong oxidizer and test for resistance to oxidative stress. We expect that under the different stress conditions and with different sexes, the methionine metabolism will be connected to these different methyltransferases. For our future plans, our goal is to eventually test all 150 of the methyltransferases. Then we will go back and retest the ones with the strongest effects. We will then use a tissue-specific genetic driver or drivers to determine which tissue or organ is responsible for the connection between methyltransferases and stress resistance. We will then test the interaction between methionine restriction and identified methyltransferases. Our goal is to determine whether this particular methyltransferases can mediate the effects of methionine restriction on stress resistance, or if it ends up functioning independently. To conclude my presentation, I would like to thank Dr. Andre Parhico, my mentor, Hillman Academy directors and team, including Mr. Solomon Lipschitz and Mr. David Boone, the TDX team, especially Mr. Andy Duncan, and Fame Academy for showing me this experience and allowing me to apply to have this experience this summer. I'm really appreciative to Hillman because this is a great opportunity that I was able to take, and I would like to explore it more in the future. Thank you very much for your time and consideration. And are there any questions?
You can go ahead and stop sharing, I think. Thanks. That was excellent. Anybody have questions? This is the first time we've heard someone, at least today, talk about using flies. So we've gone from computers and humans to mice and now to flies. well, I'll kick it off here. What you are looking at these methyltransferases, and you use the RNAi to inhibit 3 different ones. And then there's another 147 or so that you guys want to actually go and target and see how the viability responds to that. So of the 3 that you looked at, why did you choose those? Are they more, more important ones, or, what was the priority and the rationale for those versus others?
For looking into why we chose those specific ones instead of others, the goal is to test all 150 of them. So I believe that we were starting out with just those ones. I don't really believe there's a specific drive in order to choose them. We're just eventually going to test them all. So they were the ones that are readily available. And if there is more to it, I would definitely like to explore that in the future. But since I was limited in time, I was not able to completely explore the different methyltransferases or why we would have chosen them.
Got it. Because also, as you had mentioned, some of them target histones, which one might think might be maybe have more profound effects than more targeted methyltransferases.
Yes, the histones are the most common ones that I was able to research. Again, I'm not really aware if the ones we chose had specific properties, but thank you very much. I will definitely look into that in future studies.
Thank you. I'm sure there are other questions. What do you think about, why would you guys, this is not really the best question in the universe, but what do you think about working on doing these experiments in flies versus, say, a mouse model? What do you think are advantages to doing this in a model like flies?
The advantages of doing this in a model with flies is that, like I explained earlier, it's easy to tell how the phenotypes are affected and the effects of the methyltransferases in the flies because it's obviously there. And I believe in mice, it's more like the biological makeup of a mouse is more complex, like a human body is. So with the flies, it is just easier to see the effects straight away, while a mouse may take longer to see or may be more difficult to comprehend.
Absolutely.
My mentor was working with mice, but I have not been in the mouse lab.
Got it. Faster, cheaper, all sorts of things like that, I think. Anybody else? All right. Well, with that, I think, Myka, thank you, and thanks to your lab. We can go ahead and move on. So our next speaker is Nadia, who's working in the TAN lab.
Hey, Andy, I don't think that Jay's here.
Okay. I am ill prepared to introduce you. So I don't remember off the top of my head where you go to school, so I'll invite you to give your own introduction.
Sure. Good morning, everybody. My name is Nadia Lowen-Webb. I go to Barack Obama Academy in Pittsburgh. I'm going into 11th grade this year, and I've been working with Dr. Jay Tan this summer on 2 projects that I'm going to talk to you about today.
And I'll interject right here that at the very beginning of the summer, I heard from Nadia and both Jay, and they both asked me, how exactly did we pair the students and the mentors together? And I said, well, mostly there was a little bit of strategy to it. Some of it was just luck. And I think that we had probably for everybody, but really good pairings. And I think Nadia was especially impressed at how well the match was, because I think she was having such a good time. And this was on like day 7 of the program. So I appreciated that feedback, and I'm glad you guys had a good pairing.
Oh, thank you. All right, can you see my screen?
We see it.
So once again, my name is Nadia. I've been working on 2 projects this summer with Dr. Jay Tan in the Tan Lab. But for now, I'll start with telling you about my first project, which is recapitulating tau fibril spreading in cell culture. So my first project was an Alzheimer's disease project. It's likely that most of you have heard of Alzheimer's disease, but it's the most common type of dementia. It's most commonly seen in people 60 years and older, and it destroys memory and other important functions in the brain. And as of now, scientists are unsure what exactly causes the disease. So it's made up primarily of 2 things, and that's amyloid beta plaques and neurofibrillary tangles, which are tau fibrils. And tau is a protein, and fibrils, you can kind of imagine extra long strands of the protein, and then neurofibrillary tangles are those fibrils clumped together. So normal healthy tau is part of the microtubule structure, and what that is you can imagine a tube-like structure in the brains, in the cells, and it helps to transport essential nutrients from one part of the cell to the other. So what happens with Alzheimer's disease is that the normal healthy tau begins to form fibrils, and that causes the cell to begin to die. And then those fibrils leave their compromised cell and enter other healthy cells and cause the healthy tau to also begin to form fibrils. And eventually this leads to cell death. And the repetition of this process is part of the progression of Alzheimer's disease. So my project was to set up a cellular model of tau spreading. And the purpose of this is to hope that we see spreading so that we can find a way to block the spreading and in the hopes that we can find a treatment to slow down the progression of Alzheimer's. So the first thing we did was transformation in protein expression. So we have this thing called a plasmid, and that's just a circular piece of DNA, and it says digest with restriction enzyme. So we cut open the plasmid so that we can then insert our DNA sequence, then join with DNA ligase. So we close the plasmid back up, and it now encases our DNA sequence. The next thing to do is transformation. So we use bacteria, and we use E. coli to take up that plasmid so that we can then use the bacteria to express the plasmid, or excuse me, to express our protein. So after the bacteria takes up the plasmid, we grow it on a culture plate, and it creates more of itself, and we call those clones. Then we induce the bacteria to express our gene, and then the gene is transcribed into mRNA, and from there, mRNA is translated into protein, and in our case, tau protein. Excuse me for just 1 second. All right, so the next thing to do is purification. But before I talk about that process, I'd like to bring your attention to this image up here. So this gray bar is a full-length tau protein. So we purified a full-length tau protein as well as K18 protein. And K18 is just a section of the full-length tau protein. So if you imagine this gray bar is the full-length protein, then this section is K18, which is known as the aggregate domain of the full-length tau protein. So we used a purification process called his-tag purification. So the first thing we did was lyse the cells, then we bound them to nickel agarose beads, washed it, which means we got rid of some of those extra proteins that we didn't want, and then we eluted it to get our mostly pure protein. The next step was to do a process called dialysis, and the purpose of dialysis is to help remove some of those extra small molecules and proteins that we don't want that did not get removed after his-tag. And then we ran a gel to determine if this purification process was successful. So on the left is K18, and on the right is tau. And based off of this, we can determine, that the purification process was successful. So the next step after this was to assemble the fibrils. K18's ability to self-assemble is much more efficient than tau, so there's no need to use any chemicals to help it assemble its fibrils. But for tau, we used 2 different chemicals, the first being heparin, or HP, and the second dextrin sulfate, or DS. So we mixed tau with the scaffold separately, and then they shook at 37 degrees for between 3 and 7 days. So once the fibrils were assembled, the next step was to actually test if they could spread into live cells. So if you look up here on the left, this like orangish oval is what it should look like if we have just our regular healthy stable cells. These red things, we call them puncta, that tells us that they're spreading. So over here on the left is the control. These are our normal healthy cells. And then in the middle, this is K18 after 48 hours. So we assembled the fibrils, put them in with the cells, and after 2 days, you can see that there's lots of spreading. And then for tau, this is also tau 48 hours after putting the assembled fibrils in with the cells. There's also a fairly good amount of spreading. So for this experiment, we were able to successfully purify a tau protein, assemble it into fibrils, and then see it spread into live cells in vitro. So for my second project, that revolved around a protein called PDZD8. And mutations in the PDZD8 gene are in fact a risk factor for PTSD. And exactly how the protein works is unclear. But some current thoughts about it are that it's tethered to the ER and the endolysosomes. And it's believed that it transports lipids between the ER and the endolysosomes. So my goal was to set up an in vitro assay to mimic the in vivo scenario and find out if PDZD8 is transporting lipids, and if so, in which direction. So the first thing we did was PCR cloning. We got a couple of clones and we took 5 of those clones. And then through DNA sequencing, we were able to determine that 2 out of the 5 were correct. And then we can verify that through this Jello ERAM. So it says streptavidin Alexa 488. So that's looking for a twin strep tag, which PDZD8 protein has. So that's why we see these lines here, which tells us that we got what we wanted to see. So the next step was to do 2-step purification. So the first step was his tag purification, just like we did with the tau protein. So these are the elutes. And if you remember, the elute is when you get your protein and it's mostly pure. So through the Bradford assay, we were able to determine that the most protein is
Allison feeling well on the outside. A steady start for Shawnee
the bluer it shows up, the more protein there is. So obviously E3 and E4, the most blue. So we combined E3 and E4, did dialysis, and then the next step was to do twin strep tag purification because the his tag purification was not pure enough. So these are the elutes, and the most protein, again determined through the Bradford assay, is an E2. So then we ran a gel to determine if the purification processes were successful. So this is PDZD8, and through this we can see that the purification process was successful. So we have our mostly pure protein. So the next step was to actually set up our experiment and test if PDZD8 can transport lipids. So over here on the left, this lipid has a green fluorescent and a red fluorescent, but this green fluorescent is being quenched by that red fluorescent. So that means that you can only see that red fluorescent. But the idea is that when you see if PDZD8 is transporting lipids, either if it is or isn't transporting lipids, you'll see a difference or you won't see a difference. And the difference will be that this green fluorescent will no longer be quenched and you'll be able to see it. So for liposome pair 1, which is this blue one and this green one, that is mimicking the ER to the lysosome transport. And then for liposome pair 2, which is this gray one and this orange one, that's mimicking the lysosome to the ER transport. So the differences in the pairs is that one pair has, one pair of the pair has donor only, and these two up here have donor and acceptor. So that's PDZD8 taken from a donor and transporting to the acceptor. So with donor only, you can see that both of these are pretty linear. And what that tells us is that PDZD8 is not transporting anything. But up here where there's like these curves, that means PDZD8 is transporting the lipids. And since there are curves up here for both of these and flat lines down here for both of these, that tells us that PDZD8 is able to, with a donor and an acceptor, transport lipids from the ER to the lysosome as well as from the lysosome to the ER. So it can transport both ways in vitro. So to sum it up, for the Fibro project, we were able to successfully purify tau protein, then assemble them into fibrils, and then we were able to see the fibrils actively spread into live cells in vitro. And the next steps would be to hopefully find a chemical that can be used to create drugs that can help block tau spreading in the hopes of slowing down Alzheimer's disease. And then for the PDZD8 project, we were able to successfully clone, express, and purify the PDZD8 protein, and then test if it could transport lipids, and then if so, in which direction. And we discovered that in vitro, it can transport both directions. So the next steps with that would be to find out which lipids the protein is transporting and find out more about its functional relevance and lysosomal biology. So first, I'd like to thank my mentor, Dr. Jay Tan, for his support and for teaching me a lot of things this summer. I had a lot of fun. I learned so much, and for letting me work with him. I'd like to thank Torrin Finkel and the whole Finkel lab for being super supportive and welcoming. The Hillman Academy program, especially Dr. David Boone and Mr. Solomon Lifshitz, as well as Dr. Andy Duncan and Dr. Pete Alexander. And thank you all for listening to me today.
That was awesome. It was nice to get some real molecular biology coming into here. And we have a lot of protein purifications, and you're like a hardcore molecular biologist now, which is great. Who's got some questions?
I have one. So in one of the graphs you were showing, I was kind of like a little bit confused. So does the PDZ-D8, does it carry like no proteins or lipids, but then whenever it's paired with a donor, so is the donor what's carrying everything, or?
So PDZ-D8, it transports the lipids when there's a donor and an acceptor. So if it's going from one place to another place, it can do that. But if you just have your donor, then it's not transporting anything. Does that
Okay, so it needs, okay, that makes sense. Thank you.
So it needs a donor and an acceptor to be able to transport.
Got it.
In the fibril model, where you basically showed that you could purify the fibrils and then put them on your cells and you do get the spreading, how exactly does that work? So you've got your cells sitting in the dish and then you basically squirt your purified proteins on there. But how is it that they enter the cells, you know, and do what they're going to do? Because presumably that's happening with inside the cell, right?
Right. So you're asking how do they spread into the cells?
Yeah.
We don't know. That's exactly what we don't know, is how it leaves its cell and enters other healthy cells. We're not sure how it spreads.
I wonder, how did you figure out, just experimentally, the concentration of protein to put into your dish? You know, is there some threshold that you need in order for it to start to enter the cells?
So I don't exactly know how Jay figured out how much to put in, but what I do know is we would always put in 20 microliters of tau, but only 2 of K18, and
Yeah.
K18 would spread a lot with only 2 microliters, while tau would spread a fair amount with 20 microliters.
And is it spreading or just entering the cells? So for example, could you have cells where you take you've got your cells and then you add your purified protein, you let that sit for a day, and then you wash off the supernatant, and then maybe you add new cells on top of that or something? Can you actually watch it spread from one cell to the next when there's no extra protein, exogenous protein in the culture media?
So are you saying, like, if I had my cells and I put in the fibrils and then put in new cells, could I see them spread into those new cells?
Yeah.
I don't know, but I would think so, yes.
Okay. Because that's what would be happening in vivo, right? Yeah. Okay. That's cool that you established that model, and I think it really sets you up to going and like you said at the end, sort of screening through some drugs to figure out things that can stop that. Anybody else with questions? All right, Nadia, that's fantastic, and I guess we'll move on then. If anybody has a question, you can go ahead and just put that into the chat, though. So next up, we have Tomi, who is in Brian Brown's lab.
Hi, I'm Marissa. I'd like to introduce Tomi. I had the pleasure of spending this summer working with Tomi. She's a rising senior at Pittsburgh Kappa High School. She was born in Nigeria and moved to Pittsburgh when she was 1 year old. She's been part of the Artificial Intelligence Pathways Institute, as well as plays the saxophone and runs track. After high school, she plans on attending college with an engineering degree. I would like to thank everyone at the Hillman Academy for the opportunity to work with Tomi on this project. She has done an excellent job this summer, and I look forward to her sharing it with us. So you can go ahead, Tomi. Thank you.
Thank you, Marissa. I hope everybody can see my screen.
Yeah, it looks great.
Thank you. So like my graduate student said, my name is Oluwatomisin Olaore. I go by Tomi. I go to Pittsburgh Kappa, and I had the privilege of working under Dr. Brian Brown in the Brown Lab this summer. And my mentors were Dr. Mangesh Kulkarni and Ms. Marissa Thild. So first, I'll be going over the importance of the project that I worked on this summer. So TIFA is a company that develops medical devices that are derived from a new class of resorbable polymers that have been engineered utilizing recombinant DNA technology. And they reached out to the Brown Lab to help them do some research because currently TIFA's mesh are mainly used for plastic surgery, and they are trying to look at other applications. So that is what they asked our lab to do. So that is what Marissa and I were looking at, how they could use their mesh in other surgeries. So the implants that we looked at and used during our experiments were PBSM, P4HB, and the BARD mesh. So during my time at the lab, I worked on post-response implants and looked at that. And during that, a foreign body response is elicited to an implant, and an M1 and M2 are recruited, and there is an ECM response. And these responses are affected by the composition, mechanical, and material properties of the mesh that are being used in the implantations. So what happens to the extracellular matrix when a biomaterial is implanted? So during that, the ECM gets remodeled. And for the experiment, my part was going to be quantifying the collagen deposition at the site of implantation. So that is what I was looking at. So like I said before, TIFA reached out to us, and the two meshes that we were looking at for them were PBSM and P4HB, which are both thermoplastic linear polyester and are both produced by recombinant fermentation. And on the right, I have two pictures of, these are the meshes. This is P4HB, and this one over here are PBSM. And as you can see, they both look similar because they have similar properties. So what was the objective of the study that I worked on? The objective of the study I worked on with Marissa was to characterize the immune response of TIFA's mesh compared to two known meshes. And by doing that, we would be able to look at the different immune response of those and be able to come up with results after that. So what was my hypothesis for this project? So my hypothesis was, following the implantation of biomaterials into mice, there will be an observable difference in the anti-inflammatory immune cell response at earlier time points and a minimal foreign body response in P4HB conditions compared to the BARD mesh. So these were the methods that we used during this experiment. So we had implants 2 mice per group time point, and the meshes that we used again were PBSM, P4HB, and BARD. And then we had explants at day 3 and day 7. I was able to stay and complete day 7, but there are also day 14 and day 90 that my graduate student will be continuing to do. And then I worked on the histology part of this, and I looked at H&E, trichrome, Picosurious spread, and immunofluorescence. And my graduate student is going to continue on with the RNA isolation and looking at the flow. Continuation of my methods, while I was looking at the images and quantifying the collagen fibers stained blue in the images, the nuclei stained black, the muscle, cytoplasm, and keratin stained red, the background also stained red, and the hematoxylin stained the nuclei purple, and the eosin stained the cell cytoplasm pink. So these are images of pictures that I took on the Nikon, which is a microscope, and these were images of the trichrome, and over here is the BARD mesh, and this over here is the muscle. And then, like I said before, the blue is the collagen, and the little purple dots that you see are the nuclei. And this over here is the mesh that was implanted into the mice. Over here, we have P4HB. the other mesh that was used, this is the mesh as well. And the same goes for this. This is the muscle, blue is the collagen, purple is the nuclei. And then we have the sham, and the sham was without the mesh. So this was just surgical without. So this has no mesh in it. So this is why this doesn't have these big mesh fibers in this, because this wasn't implanted. And over here we have our PBSM. Again, we have our mesh that is implanted and our collagen and our nuclei. So this is H&E. These are images that I took from the slide scanner, and over here is the sham. Again, this doesn't have a mesh, but over here we have our BARD, and these over here, these are all meshes that were implanted. And in this, the hematoxylin stained the nucleus purple color, like I said before, and the pink was the eosin. We have our P4HB here again and our PBSM. So what were our total cell counts for the H&E? So whenever I did this, I used QPath, which is a software system used to analyze pathology, and it helped with our cell counts. So for the BARD mesh, we had a total of 11,728 detections. For our P4HB, we had 15,966. For the P4HB2, we had 8,161. For the BARD2, we had 11,664. For the PBSM1, we had 5,144. And for the PBSM2, we had 18,762 cells detected. Next, I have our Trichome images. So basically the blue here is the collagen, and the little purple dots are the nuclei, like I've said before. And these are just the mesh that were implanted. Again, we have our BARD, P4HB, and PBSM. And we were unable to get cell counts for these yet. Well, before I left the lab, we were unable to because of some technical difficulties that we weren't able to figure out yet. So we weren't really able to get the specifics to be able to count the cells well enough for the project. And the last one I had was pick a serious red. So the red is the collagen, which is what we were looking at. We have our sham again, our BARD mesh, the P4HB mesh, and our PBSM mesh. And again, the same thing as the trichome, we weren't able to be able to count these cells yet, so we weren't able to do that. And during my stay at the lab, this is not really important to my project, but this is just something fun I got to do with my graduate student. This was a CD11B immunofluorescence stain. So these were the BARD mesh, we had the P4HB again, and we had the PBSM again, and these were the mesh. And basically the red that you guys can see tagged the CD11B expressing cells, and the blue that you guys can see stained the nuclei. And since we weren't able to finish the project, because this is a continuation and it's taking longer than I could have stayed at the lab. So I don't really have any conclusions because my graduate student isn't done yet. But if I was to continue to stay in the Brown lab, I would compare my day 7 results, cell counts, with the other time points. And then we would be able to draw conclusions from that because we would be able to compare the different cell counts that we had and see which one was more effective. And for my acknowledgements, I would like to give a big thank you to Dr. Byron Brown for letting me stay in his lab, Dr. Kulkarni Mangesh for helping me while I was there, Miss Marissa Thilt for being with me since day 1 and helping me and letting me do her project with her, the entire Brown lab for everybody welcoming me since I was there, Dr. David Boone from the Hillman Academy, and also Dr. Solomon Lifshitz from the Hillman Academy. And I would also like to thank Dr. Andrew Duncan from the TechX Drive site for helping us while we were working on our presentations and preparing. So thank you, everyone.
Great job, Tomi. And those are just beautiful images that you've captured this entire summer. A lot of different types on there. Anybody have a question? I'll kick it off. You showed, I forget exactly, it was one of your earlier stains when you were quantifying the number of cells, and you had some pretty big differences. Like I remember, like there was 18,000 cells in whatever area, which was quite a bit higher than you had seen in some of the other groups. So what do you think is happening with this the mesh from the company? What's your prediction? Like, how is that really affecting this foreign body response?
Well, I did notice that as well. And one thing that did have to do with that is that some of the image quality from the slide scanner didn't turn out as good as others. So QPath wasn't really able to detect those cells as well as others. And we tried to look at that and fix that, but we're unable to. So that's why some of them were like as low as 8,000 and the other one that was as low as 5,000, because some of the other pictures, even though they were from the same mesh, had higher ones. Like the PBSM had 5,000. The PBSM2 had 18,000. So some of them were the quality of the images. So I guess if we wanted to try and redo those stains and then slide them again through the image scanner, we'd be able to get better cell counts.
Got it. Okay.
I also have a question. So whenever you're talking about the different meshes, like P4HB and PBSM, I think, what are some of the differences between the meshes that could kind of like affect your results, if that makes sense?
To answer your question, I'm not exactly sure. There aren't really much of a big difference between P4HB and PBSM. We are trying to compare those two to BARD, and Marissa was also looking at alloderm, but we weren't really able to add that into my project. So those are the two that they were comparing them to, because P4HB and PBSM are pretty similar to each other. So they would probably have like around the same results.
Okay, thank you.
And you didn't get too much into this in the presentation, but I think one of the ultimate goals is kind of looking at the macrophage phenotypes that are infiltrating. Is that right? Like going into the M1 and M2 and whether you're getting remodeling and so forth.
Yes, we did. I did read about M1 and M2 over the summer, and that was also part of this project and looking at that, but I didn't really look much into that, but that was part of my hypothesis.
Do you have any, can you remind everybody about M1 and M2 and maybe what you might expect to happen with those?
I'm sorry, could you say that again?
Can you just remind us all about M1, M2 macrophages and maybe what you would expect to happen?
Yeah, M1 macrophages typically have a pro-inflammatory response to the immune response, and then the M2 macrophages usually are the opposite of that and aren't really inflammatory. So those were the two different between them. So in my hypothesis, we had said that, I believe we said that it was going to be an anti-inflammatory immune cell response.
Got it. I wonder, sort of putting your work together with Madeline's work, where she was, they're having these probes inserted into the brain and they're looking at the foreign body response there. I know that in sort of the whole bioengineering field, you're looking at, people are thinking about ways you can have these meshes or foreign devices that can actually direct the outcome of the cells that or change the way the cells are responding to that foreign body. So maybe you guys need to work together and figure out how to make better probes for the brain too.
Yeah, we could definitely look into that in the future.
Next summer, that's what you can do. All right, any other questions, you guys? Well, thank you. That was excellent.
Excellent work, Tony.
And we're gonna move on now to Enysah, who is in the Gurkar lab.
Good morning, everyone, and thank you for joining us. It's with great pleasure that I introduce Enysah Roberts, who is a rising junior at the Ellis School and now a member at the Gurkar lab. It has been so much fun to have Enysah in the lab. She is so motivated and so driven, and she is such a great team player. I think her spirit of being a team player comes from her love and dedication for sports, especially basketball. And she wants to grow up and be a surgeon. And I'm pretty sure she will get there because she is so driven. As you'll see from Enysah's talk, we work on a really microscopic worm called C. elegans. And Enysah had this opportunity to work with a very talented lab manager in my lab, Suhao Han, Dr. Suhao Han, and I think she's here too. And Suhao mentioned to me that it took her months to really work with C. elegans and to make sure that she could pick them and transfer it without causing harm to the animals. But Enysah learned all of this within a few days, within a couple of days actually, which to me is amazing. So I'm really hoping that Enysah can come back and be in the lab next summer or even when she's off to college. So Enysah, you're welcome to the lab anytime. And without further ado, I'm really looking to listen to what Enysah did during the summer. Thank you for having us.
Thank you for the introduction. Hi, everyone. I'm Enysah. And this summer, I worked with Dr. Suhao Han and Dr. D.T. Gurkar from the University of Pittsburgh. And today, I'm going to be presenting on the cell-autonomous and non-autonomous role of P53 in lipid metabolism. So the world population is aging at an alarming rate, and older adults are at higher risk for COVID-19, Alzheimer's, and heart disease. And older adults usually lose body fat, making them frail and susceptible to diseases, falls, and hospital readmission. And my project was to see if there was a way to prevent this. And this picture here shows, like, as the elderly population continues to increase, the costs associated with the elderly population puts a tremendous pressure on our economy, and this calls for a demand in a healthier, older population. So in the lab, we use C. elegans as a model system, mostly because several scientists working on C. elegans have gone to make huge discoveries and won the Nobel Prize. And overall, they're really great to work with in the lab. And C. elegans are microscopic transparent nematodes that live for about 2 to 3 weeks. And their quick life cycles gives us a good amount of time to analyze these worms and also to create an opportunity to study human aging without having to rely on the relatively long lifespan of mice and humans. And C. elegans are also great to work with when studying human biology because they share many biological characteristics with humans. A hallmark of aging is DNA damage, and we are all exposed to DNA damage constantly. For example, walking out in the sun, eating burgers and fries and junk food, and even day-to-day DNA replication errors. And to study how DNA damage drives aging, we use C. elegans that are deficient in a DNA repair protein called ERCC1. And the loss of the DNA repair protein makes humans, mice, and even worms age in an accelerated manner. So previously in the Gurkar lab, they observed the loss of ERCC1 leads to the loss of body fat consistent with aging. And so in this picture on the left, you see 2 sets of worms, both stained with the red pigment called Ororedo. And so this Ororedo primarily stains body fat in C. elegans. And the worms on the left of the picture are N2 worms, which means they are wild type. And the worms on the right are ERCC1 worms, which means that they have a deletion of the ERCC1 gene. And so the chart on the right measures the amount of fat in each genotype of worm. And you can see that N2 has a greater amount of fat compared to the ERCC1, which is represented in the chart. And this point is evident in the picture as well, because you can see that N2 has a lot more red. And this just shows that due to DNA damage, there is a reduced amount of fat indicated by the lack of red in the ERCC1 worms. And so in conclusion, this accumulation of DNA damage leads to a loss of body fat. So P53 is a DNA binding transcription factor that is activated by DNA damage. and it can then regulate genes responsible for cell cycle arrest, DNA repair, senescence, and apoptosis in response to DNA damage. And what this means is that P53 acts as a light switch. When DNA damage occurs, it is turned on and expresses specific genes for that certain task, and it is turned off when it is not needed. And P53 in humans is CEAP1 and C. elegans, so they're kind of parallels of each other. And so the goal of my project was to identify whether P53 CEAP1 plays a role in fat metabolism in a cell-autonomous or non-autonomous manner. Cell or tissue autonomous means that cell or tissue is self-governing. It only relays information to that certain area it needs to, which is inside its own cell. And cell or tissue non-autonomous means that it can relay information to other cells, allowing for genes to be expressed in one cell to affect the physiology of other cells. And it has the ability to affect processes outside of its own cell. And so using the analogy of a light switch again, cell autonomous is a light switch in the kitchen that can only turn on and off the lights in the kitchen. And cell non-autonomous is Alexa, which is in the kitchen. It can control the lights in the kitchen, but also has the ability to turn on and off lights in the dining room, in the living room, and even the upstairs hallway. And so a part of my project was figuring out how P53 CEAP1 functions in fat metabolism. Does it function in a cell autonomous manner or a cell non-autonomous manner? And in order to see this, we would need to generate tissue-specific RNAi worms. And what this means is that we would feed worms a bacteria that would target and silence P53 and CEAP1 when consumed by the worms. And the reason it's called tissue-specific RNAi knockdown of P53 CEAP1 is because when the P53 CEAP1 RNAi is consumed, the gene will be silenced in that particular tissue area. And so the tissue-specific RNAi strains we generated were ERCC1, RDE1, which are germline-specific RNAi worms, and ERCC1 PPW1, which are somatic-specific RNAi worms. And to sustain body fat in these worms, we performed the following experiment where we got eggs from adults that were synchronized, which means that they were all at the same age, and we let them develop into young adults. And then we transferred them to RNAi plates, and I picked new worms every day to make sure that they had food and that they weren't starving. And then I let them develop into day 7 adults or older adults. And then we would stain these these worms with the red or redo dye, and then look at them under a microscope. So the first thing I had to do was check that my controls work. And this is where CEAP1 is knocked down in all tissues. RNAi is a process in which you silence a particular gene. And in this case, we're silencing CEAP1. And now that we have silenced this gene, the ERCC1 worms have restored their fat. And you can see this in the chart on the left. It's quantifying the amount of fat in the worms. And with control RNAi, there are no genes being silenced. So N2 has a nice amount of fat, and the ERCC1 compared to the N2 has very little. But on the right of the chart, when CEAP1 is being silenced, you can see that N2 worms have not changed that much in fat, but the control RNAi worms with the ERCC1 and CEAP1 mutation have regained most of their body fat. And you can see this also on the right, comparing these two pictures, that the ERCC1 mutants with CEAP1 RNAi have more red than the ERCC1 mutants. And so in the future, we are going to knock down CEAP1 in a tissue-specific manner to determine where and how CEAP1 controls fat metabolism. And for my acknowledgements, I would like to thank the Gerker Lab, especially Dr. Aditi Gerker and Dr. Suha Han for making my experience in the lab worthwhile and just being patient with me in the beginning when I knew very little and just helping me out through this program. I really appreciate it. And thanks to Dr. David Boone and Solomon, the University of Pittsburgh, the Hillman Cancer Center for providing me with such an amazing opportunity to do research this summer. And also a great thank you to FAME for introducing me to this opportunity. I had a great time this summer and I can't wait to do it again. Thank you.
Enysah, that was fabulous. That was probably the best description of cell autonomous and cell non-autonomous I've ever heard. So good job with that.
Thank you.
You guys have any questions? Okay, to kick it off, just big picture wise, can you explain the relationship between P53 and ERCC1?
Yeah, so P53 is just a gene that allows for protein coding to happen in relationship to DNA damage. And so ERCC1 is basically also a protein coding gene that helps DNA repair. So like the deletion of CEAP1 in the ERCC1 worms helps to restore the fat. I hope that makes sense.
Yeah, I'm wondering, and I think I know the answer, but I'm not entirely sure. Is ERCC1 a P53 target? So P53 acts as a transcription factor, right? Yeah. And then does it induce expression of ERCC1?
Yes.
Okay. Okay, so when P53 becomes activated, you get ERCC1 expression, right? Anybody else here? Did you have, so you looked at fat accumulation in these worms, and you've got beautiful images on that. And I remember at the beginning of the summer, we talked a little bit about where was the fat accumulating. And I told you that I always have a hard time telling the head from the tail of a worm, and you explained how you could determine that. Do you see any patterns of the lipid accumulation or the oil red O staining that you could make a hypothesis about what you would ultimately see when you do your tissue-specific knockdown of ERCC1? What are there tissues you think are more important than others?
Well, in the intestines, that's where most of the fat is stored. So we're hoping that when we knock down the CEAP1 gene in the intestines, that it will be cell autonomous.
Got it. Okay. So you think that's the major organ, I guess, that's affecting that?
Yeah.
Did you see anything with aging? Or, I mean, I know you didn't really look at aging, but was any, did you see any like more death or anything like that with the knockdowns?
No, because the worms primarily died after we stained them.
Fair enough. Sorry. Yeah, you're right. You're right. Okay, got it. But everybody made it to whatever time point that you were trying to harvest them at.
Yeah.
Got it. Okay. All right, everybody. That is Alina here? Alina is the one who was sick this morning. And I guess we were going to try to punt her to the very end, but she may still not be feeling well. You guys did a great job. I was very just delighted and proud to hear how much you've done this summer and how well you articulated your experiments and the big picture and all that. I want to thank all the mentors again for your hard work and dedication for hosting the students. And then this afternoon at 2 o'clock, there's a closing ceremony and everybody's invited to attend that. You'll notice that I put a link into the chat box that has the schedule with all those links on there. So students will join the Zoom link, parents and friends and whoever else can join the YouTube link. I see right now that there's a question about what's going to happen to this recording. I'm going to turn it over to Hillman Academy, to David Boone and Solomon, and I think they can decide how to distribute that. If you don't get a satisfactory answer from them, then you're certainly welcome to contact me and we'll figure out what to do with it. I hope that helps. With that, thanks for coming. And can I just ask all of the students to stick around for a minute at the very end, and we're going to take a group photo by Zoom, and it'll just take like 1 minute. So thank you, guys. All right. We've got most people here. Looks like a few people got a few stragglers who aren't students. And we lost a few people. Well, all the students, if you guys could just make sure your cameras are on. Griffin, Myka.
Hold on a second. My nose
Okay.
is bleeding.
Oh gosh, okay. Yeah, we'll give you a minute for that. That's fine. So you guys did great. How do you feel about that? Good. Is anybody gonna, I know that Felipe is going on a big trip tomorrow. Is anybody else gonna have a vacation or anything coming up before school starts?
Yeah, I'm actually, I'm going to Chicago tonight, which is pretty cool.
Oh, good, good.
I have a college trip planned to University of Michigan, so that's cool, I guess.
Yeah, that is. Ann Arbor is beautiful. Yeah, we definitely don't want the bloody nose in the picture. Today becomes much easier with the way Hillman Academy is this year, because as I told you before, we normally do these talks in the morning, then we do kind of a lunch in one of the break rooms, and then everybody would go to the Hillman Cancer Center for a couple-hour poster session, which it's a lot of fun to do posters because then you get to present your work to even more people. But I'll tell you, at the end of the day, I'm not even the guy presenting, but I'm exhausted. It's just a lot. Priya, that's how it was when you were, when you did it, right?
Yeah.
Yeah, that was a long day, wasn't it?
Yeah, the posters were definitely really fun though, because you got to re-articulate yourself every time someone new came up to you. You could like re-explain things in a different way. It really helped you understand what you were talking about.
That's a great point. You guys ever hear about like comedians who are gonna like get their show fixed? You know, they're trying to figure out all their jokes, so then they go and they give all these shows and they figure out what jokes work and what don't before they go and do a big Netflix special or whatever. That's exactly what a poster session is, because you talk to people for 5 or 10 minutes, and if you get a sense that, oh, that didn't go quite right, you just change it right then, and you keep honing your message like that. All right, Myka, you're back. Okay, let's see here. We are gonna do a screenshot. So I will say, everybody say molecular biology right now, or just smile, whatever you want. All right, we got one. And now let's just do one more. Okay, so whatever, just give a big smile. Got it. All right, that's fantastic. I will see you guys at 2 o'clock. All right, you did a great job. I'm proud of you.
Have a good day.
See you.
Have a good day. Thank you.
Thank you.
Thank you.